This is the podcast for the journal Neuropsychopharmacology. I'm Cynthia Graber. Researchers are interested in understanding the biology of why some people are more likely to overconsume substances. Some substances are difficult to study. People might not admit to illegal substance abuse or to how much alcohol they drink. But Americans are more likely to accurately recall and share how much coffee they drink, which is related to how much caffeine they consume. And so a team of researchers paired up with the company 23andMe to try to understand genetic differences among a large set of people, to try to tease out any genetic similarities that could be correlated with coffee consumption. Sandra Sanchez-Roige is an associate professor in the Department of Psychiatry at the University of California, San Diego, and she's one of the study's authors.
The reason why we partnered with a genetic company like 23andMe is that we are scanning the whole genome. Well, in truth, we don't scan the full genome because we know very well the structure of the genome, so we don't need to scan the full 3 billion base pairs. But we scan millions of sites on the genome. These sites have very small effect sizes. And so we are now facing a statistical problem of multiple testing correction. Each variant has small effect sizes, and then we're testing millions of them. So clearly you need a very large sample size if you want to detect and believe that the results that you're getting are statistically significant.
And this is why they needed to work with a data set as huge as the one 23andMe has amassed. Abraham Palmer is a professor and vice chair of basic research in the Department of Psychiatry at UC San Diego, and he's another of the study's authors. He says the gold standard double-blinded study, which he's done in the past for caffeine, that won't work for this type of genetic study.
The trouble is you'll never get 100,000 or 1 million people through a paradigm like that. And so what we've done moving to study designs like working with 23andMe is we've sacrificed that kind of quality of phenotyping. Obviously, we're asking people, they might be wrong. People drink different kinds of coffee with different amounts of caffeine in it. We're not making any attempt to correct for body weight, right? So there's many, many layers of sort of confounding or muddying factors here. And yet, with the very large sample sizes, it actually works out to be better for genetic studies.
In the end, they analyzed data from the genetics of more than 130,000 individuals. And as part of a survey overseen by 23andMe, they were able to ask those people how many cups of coffee they drank and then try to pinpoint what could be relevant similarities. So what did you find?
We identified that how much coffee you drink can be linked to your genetics, which some people find surprising. But it is important to note that there's not a single gene that we identified. Now, I think that because of candidate gene studies were initially dominating research, it's not surprising that we've come across headlines saying that there's a gene for X. And of course, there is genes that influence how much coffee we drink, and this is what we found, the usual suspects, genes that indicate how rapidly you can metabolize caffeine, like remove it from your body. That is not surprising to think that if you can clear it out very quickly, then you may need to drink more coffee in order to have the same effects of feeling alert or energized. But there's many other genes and genetic variants that influence how much we drink.
Broadly, we could think about genes as having either pharmacokinetic or pharmacodynamic effects. So pharmacokinetic effects would be the ones that clear the caffeine, that metabolize the drug. That might mean that you need to take more drug. It might also mean that you have fewer negative side effects. So for instance, people that clear the caffeine rapidly probably can go to sleep more easily at night. People that have a very hard time clearing the caffeine may find that it interferes with their sleep and they may choose to drink less coffee. And then the pharmacodynamic ones, which I feel, to my mind anyway, are more interesting, would be the ones that influence things like the subjective experience of the caffeine. So some people, and I have never understood this, feel that caffeine makes them anxious and jumpy and uncomfortable, whereas other people like myself just really like the way that it makes them feel and get that kind of energized feeling and at least the illusion of having greater focus. And so therefore like it. And those are not probably due to the metabolism, or at least that's not my belief. Those probably have to do with how the actual drug changes the biology of your brain.
So you're saying that you found similarities. You're not yet sure what those similarities actually do, but they might be related to those pharmacodynamics?
Yeah. And that's where maybe we're learning something new about biology. So the metabolism of caffeine, I think, was reasonably well understood before we did this. And so we found genes, I think one of them is CYP1A1, which was well known to be a gene that encodes a P450 enzyme that is involved in metabolizing caffeine. So that's not a new discovery in our study. We would have expected that if variants existed in the population for that gene, that they would influence caffeine metabolism, and we think that would influence caffeine consumption. Other genes identified by the GWAS are not believed to be involved in the metabolism, the pharmacokinetics, and therefore we think they may be more pharmacodynamic. And one of the things we would love to have, but don't currently have, is data about the subjective effects of caffeine. So we ask people how much they drink, and I'm willing to posit that people drink more or less coffee partially based on how it makes them feel. But we didn't ask them how it makes them feel. And I would love it if we could do follow-up studies in this cohort or another cohort where we could ask about caffeine-induced anxiety, caffeine-induced euphoria, caffeine-induced sleep disturbances, the things that probably are motivating people to drink more or less coffee. Unfortunately, we don't have direct data about that in this study.
In your paper, you compared your results to data from a UK biobank because you wanted a still greater sample size. What did you find out?
Surprisingly, we discovered, as we did that meta-analysis, that caffeine consumption in the US cohort from 23andMe and caffeine consumption in the UK cohort from UK Biobank didn't behave as if they were the same trait. And that seemed surprising to us because isn't coffee is coffee, but maybe coffee isn't coffee. A couple things come to mind. One is that in the 23andMe study, we had asked about coffee and we specified caffeine-containing coffee, caffeinated coffee. In the UK Biobank study, we didn't, of course, control the question that they asked. They had asked about coffee in a way that allowed for both caffeinated and decaffeinated coffee. So really, the phenotypes weren't exactly the same. That's an undesirable feature of the two data sets. Moreover, we know that they love to drink tea in the UK, and that one of the really cultural differences between the US and the UK is the meaning of drinking either coffee or tea, both as a caffeine source, but also more broadly as a sort of a cultural ritual, behavior that people do. And we believe that some of the differences that we observe between the US and the UK, and again, it's difficult to test this directly with data, but we believe that some of those differences are driven by both the differences in the way the question was asked and the failure to account for other sources of caffeine, which is probably more important in the UK.
So there were a lot of hints to indicate that potentially we should have not combined the two data sets in spite of power. And I think this is one of my favorite results of this paper because it provides a cautionary tale on combining data sets moving forward.
Interesting. So one of the questions you started with has to do with what we can learn from this about overconsumption in general. Do you have any clues?
I think more broadly, we're interested in learning about overconsumption. This is a behavioral trait that cuts across many different substances. Of course, coffee and caffeine, which is not what we studied, it's a proxy for coffee consumption. It's not an illegal substance. We don't mean to say that the people that drink coffee may immediately consume other substances. This is not what the study was designed to do. But we find that the biology that underlies this overconsumption cuts across different substances and potentially other food-related behaviors and other behavioral traits. And so we hope that by learning the genetic basis of this overconsumption, in this case of coffee, we may learn about the biology of why people may also overconsume other substances. And in fact, when we looked at genetic correlations, we identified that genetic variants that are implicated with coffee consumption are strongly correlated with genetic variants that are associated with consumption. It's mostly consumption phenotypes. It's not clinical diagnosis of substance use disorder, for example, but it can give us clues about what we deeply care about, which is why some people consume substances in a way that it leads damage to their personal lives and the families around them.
This is the podcast for the journal Neuropsychopharmacology. To read the article discussed in the podcast, go to www.nature.com/NPP. I'm Cynthia Graber.